All right, thanks very much. I'd like to thank the previous speaker first for saving me a lot of time of describing CAFA. And I'd also like to thank the organizers for inviting me. And finally, I'd like to thank Leila from yesterday, who gave me a great idea. You know, she said how one important component in talking about science is how we got here. So maybe I should talk about this a little bit, because it actually, as you'll see, it dovetails very well with what I'm doing these days. So, you know, when I pretty much started as a structural bioinformatician. This is me at the end of my PhD. I'm still smiling, and I was thinking about things regarding to sequence to structure predictions and how sequence affects structures and things like that. Then during my postdoc, I went more into the idea of okay, a structure is interesting, but really, what do these proteins do, and how do we predict function? And I really wanted to learn about that. So yeah, I read a few papers that weren't enough, so I thought maybe I should go to a conference with people talking about that. And I couldn't find such a conference, really, so I started to organize one. So in 2005, I started organizing what was known then as a function SIG, special interest group, now known as a function COSI, COSI, Community of Special Interest, and that has been running for 20 years now within ICB. So I'm very grateful for ICB for giving me a platform to do that. And the next one is going to take place in Liverpool during ISMB 2025 in July. And during that time, you know, we talked a lot about function and function prediction, and you heard a lot about that from the previous speaker. And that is something that I really got into. And in 2010, we founded CAFA, which was described to you as this competition of protein function prediction. We had 5 rounds until now. We run CAFA every 3 years or so. And now, you know, when I'm kind of older, I'm starting to think about what is function. And a lot of people here talked about protein function and predicting and so on. They gave different definitions and different takes on what function is. They're all correct and all true. It really depends on the type of functionality you're actually looking at. In this particular talk, I will narrow down the definition to what was said before, and we'll talk about representing protein function as the gene ontology. So an ontology is really a great mechanism of really representing anything. We can have a conference ontology, right? We have attendees, and we have speakers, and we have organizers, and we can connect them in this kind of a graph thing, like you see here. So these are ontologies for describing functions. Only functions have different aspects, right? You can have this is an ontology describing pyruvate carboxylase, which is both an enzyme that turns pyruvate into oxaloacetate. That's you can call it the molecular function, what it actually does, but it also participates, it's also the entry from glycolysis to the TCA cycle, and thus participates in a process. And then the biological process is represented in this other graph, which is the biological process ontology. So this is these types of graphs, they're hierarchical, but they're not trees, because a tree, each child has to have only one parent. These children have more than one parent because you can participate in more than one biological process, for example. So these are directed to the cyclic graph, or DAGs. So the problem of protein function prediction really can boil down to something like this. You take the sequence and you predict a subdag or a collection of subdags, a very large collection of subdags sometimes from it, all the processes that are associated with it. And one specific rule that I need to introduce here is that if you predict any kind of node on the subdag, that automatically implies all its parents, what we call propagation, or the true path rule. Of course, you want to add as much data as you can. You know, if you find data on the structure or point direction or expression, that helps a lot in the prediction. Recently, embeddings, which is, you might think of them as a kind of a representation that capture one or more of these aspects together and put them in, have become incredibly popular, and we heard about that in the previous talk also. So how does CAFA work? CAFA is really mostly a prospective challenge, which means that in the beginning, what we have during the prediction phase, we have a protein for example, a protein for which we know nothing about anything in the different Go aspects, the biological process, the molecular function. There's also a cellular component aspect. I won't get into it. Basically, where the protein is also important, also an important aspect of the function. But let's say we have this one protein here. We don't know anything about it. Then over time, we get to know things. Papers get published. Biocurators read those papers and put the data into the database. Thus, we get the phase called annotation growth. Suddenly, this protein gained annotation in these two other aspects. So if somebody predicted in the prediction phase, in the blue phase over there, is there a pointer here? If somebody predicted something here and they got some of these nodes right, they'll get a certain score for that during the assessment phase. So this is really how CAFA works. So here we have the history of CAFA over the years. Like we said, we started sometime in 2010. The blue is the prediction phase, red is annotation growth. CAFA 4 started in a very unfortunate year, 2020, both because of COVID and because everybody was moving around different universities at the time from the people who were organizing CAFA. It took us a very long time, but we're finally putting the paper together. So we published 3 CAFA papers. We have a 4th that's already in a good shape in a preprint. So that was interesting. When we started CAFA, we had 30 participants. In CAFA 2, that grew to 56, then 68, then 70. It kind of plateaued. And we had certain challenges. We basically didn't have any funding towards CAFA 4. We were basically running on fumes and various IDC accounts. It seemed like our engagement was plateauing. We were really limited to academia, to groups who were doing bioinformatics predictions. And 70 is a nice large number, but we want to get more people. And we were kind of disconnected also from the machine learning field at large. So in contrast, CASP was just getting AlphaFold to work, and that came totally outside the ecosystem of academia. That came from Google DeepMind. So we were trying to engage more. So luckily enough, we were approached by Kaggle, which is now a Google subsidiary. And Kaggle is this platform in which basically it's a data science platform, machine learning platform, and they also run a lot of machine learning competitions. And they wanted to run CAFA 5 there. And we said, yeah, okay, we'll do that. And they helped us a lot putting together the competition, helping us put our assessment software up there on their on their platform, advertising, and so on and so forth. And especially, they provided prizes, a total of $50,000 split the way you see here. So the top 6 places got really nice prizes. That recruited in a lot of participants, over 18,000 participants into 1,600 groups or so. The numbers come later. How did we run this? We had a potential evaluation set of 141,000 proteins from 19 species. And we get the training set of those proteins that had a known function in at least one gene ontology. You can see there's a certain overlap here. The leaderboard contained held out annotations, really only just 78 proteins. And these 78 proteins are proteins that were supplied to us from the European Bioinformatics Institute that they did not put in UniProt specifically because we wanted them to be in CAFA so that this could run on the leaderboard and people would know what they're doing. So CAFA turned from a prospective challenge into a challenge more like a hideout, at least for the purpose of the leaderboard. However, when we evaluated, we did use an accumulated set, which was a bit larger, just under 500 proteins. So that was nice. The nice thing about Kaggle is that they're a very friendly and interactive platform. We got a very dynamic leaderboard. Things went up and down there all the time. People were talking a lot with each other. Kaggle has these rules of competitive openness. So you have to share data on the platform if you're using it. You cannot sort of form these cliques behind the curtains. That's illegal. There was a lot of code sharing going around. There are webinars, fantastic webinars organized by some of the groups, and I personally learned a lot from those. So that was a lot of fun. And if a participant reports some sort of error, that gets immediately amplified, of course, and then we have to update the public evaluation code and we scored the leaderboard and so on and so forth. So there's not a lot of that, but that happened, I think, once. Finally, the participation surpassed previous participations in less than a week. We got 1,625 teams that are participating from around the world. So you can see both the reach of Kaggle and, of course, the incentive of the $50,000 cumulative prizes helped a lot here. And this is the timeline for the CAFA Kaggle annotation, CAFA Kaggle competition launch. We launched in 2023. Closed the prediction phase in August 2023, launched in April, closed in August, so the traditional 4 months. And finally, the evaluation took place after a while in May, and then again in August. But in May, we actually gave out the prizes. So these were the top teams in CAFA. It was really interesting. Some of these are classic CAFA competitors. Some of these came from completely outside the field. There was one guy from, I think, Toyota Research or something. Another group that came from actually one place where I postdoc, the Burnham Institute. They were biologists with almost zero knowledge of machine learning, but they did incredibly well. So it really shows that the diversity of thought and skills really helped a lot. And you can see that, and they're supposed to also provide solution write-ups. And you can see that here. Do I have some more time because we started a bit late? Or okay, because you're standing. So okay, thanks. I want to keep time. All right. So let's go to the evaluation a little bit. First idea: whenever you evaluate machine learning, you got to understand what your baseline is, right? Because, for example, in CAFA 1, we evaluated. When we started to go through evaluation, we knew nothing. We didn't look at the data, which was our first mistake. And when we looked at the data, we realized that 30% of the proteins have protein binding as their term. So if I write a Python script that says print protein binding, I have 30% accuracy, and I don't even have to have input. So we kind of threw that away in CAFA 1, and we learned from our mistakes, and we tried to make the data a little bit more balanced. Still, one of the baselines that we use is, of course, what we call the naive baseline, where we predict all the proteins function according to the annotation frequency in the training data. So just doing a precision recall curve based on that. Another baseline that we use is BLAST, because everybody uses BLAST. So if you use BLAST for predicting function by homology, then you want that. Uniprot non-experimental, which is assign all the annotation with non-experimental evidence in the training data. And finally, because everybody's using embedding, we also use embedding similarity as a baseline, because that's kind of a frequent way of doing things. And as it was shown here, for LLMs and for embeddings, just doing embedding is not enough. I'm going to show just really 2 or 3 results here. These are the results from the no knowledge competition, meaning basically we don't know anything about the protein when we start off with. So that's the blank graphs up top, and then we learn something filling in the blue circles, and you see the precision recall curves down there. The dashed lines are the baselines, and the colored lines that are above them up to the right, that's your regular precision recall curve. You see that they are doing better than the baselines. This is the molecular function ontology, the biological process ontology, and the cellular component where it's at. This is something we call the limited knowledge. We know something in 1 or 2 of the gene ontology aspects. We don't know anything about the third. The prediction assessment is on the third. Finally, we introduce something new that's called partial knowledge, where we might know something about the protein, but if we gain more knowledge, that is assessed as well. That's the hardest, as you can see. The precision recall curves here are flatter. So that's a very hard problem to solve, and we just introduced it in CAFA 5. And we're going to do it more of that because it's hard and it's fun. The one thing that CAFA 5 gave us is much better results overall. These are box plots to show the distribution of the results of the Fmax that was talked about before. In CAFA 2, this is how well they did, for example, in limited knowledge, CAFA 3, CAFA 4. And you can see CAFA 5 is usually much higher than the rest. And this is for the top performers, I believe, for the top 50. So in the different tracks, no knowledge, limited knowledge, partial knowledge, mostly CAFA 5 performed better. How do we compare CAFA 5 to previous CAFAs? Well, we basically run on the same, on a subset of the same proteins for everybody. Okay, so I'm finishing up here. Takeaways from CAFA 5 experiment. We record growth in participation and in skill diversity, which improved things a lot. Significant improvement in the prediction, especially in biological process ontology. Diversity of approaches, spur combined models using promising idea from each methods. And the partial knowledge is a very challenging setting with opportunities to harness known function for conditional predictions. And I'd like to thank everybody, but especially Parnal and Anne, who are here in my lab, and who are here for my lab, the people from Northeastern University, my colleague Pedja and Clara, who's the graduate student of Pedja that really run everything, or almost everything on CAFA 5. Damiano and Silvio wrote the CAFA evaluator, which was talked about before, and you're welcome to use it. And of course, the CAFA participants. And thank you all for listening.
I think I might finally begin to understand CAFA. No, this was the first year where you had, because of the leaderboard, you had a set of proteins about which something was already known, but that information was hidden.
Yeah.
So which meant that people
didn't know which proteins those are. They knew the proteins are there, but they didn't know which subset of the proteins are the one that we were assessing on.
Yeah. So, I mean, my question, let's see if I can get this straight. You have 78 proteins, which could have any amount of, they're not a random sample. They're what was sort of available at the time. Do you see people, when you looked at the final sample you evaluated on, was there any kind of bias that resulted from the 78?
There was definitely a difference. I mean, there was one group that hopped from 30th place to 4th place. There was definitely a difference. And we did say in the beginning of the CAFA evaluation that, the results on the leaderboard at the end of the prediction phase are not the ones that we assess on. We assess on a larger cumulative classic CAFA thing. That was made very, very clear in the start.
Well, I was more interested in the statistics than I wasn't worried about fairness. I was interested in how the statistics worked out that way.
It's probably somewhere in the paper. I'll have to look at that. I don't know. Yeah.
Okay.
Yeah. All right.
That was enough. Thank you.
All right.
I was just wondering if you or any of the participants like accounted for other aspects of the Go information, like the evidence codes and the binding partners and
Yeah, I didn't say that. Yeah, we did.
properties.
So, yeah, I didn't say that. So what is a true function? How do you know what a true function is in the first place, right? So the idea is that we use the evidence codes just to get everybody here on board. Evidence codes are basically, how do we know the protein does this? So one way is electronic. Just, I blasted it and I found a similar protein, therefore I infer that. That's pretty weak, and that's called electronic annotation. However, if you have a paper in which somebody did an experiment, that's a much stronger evidence code, and that's called an experimental evidence code. So the idea is that that known functions are known because we have an experimental evidence codes for them. I kind of omitted that part, but that's how we know that the function is correct. So that's a very important part in which we say, okay, we don't know anything means there might be blast evidence, but we don't consider that strong evidence. But if over time some experimental evidence accumulates, that adds that little blue dot in the graph, and that's how we get our data points.
Did any of the participants incorporate that into their training and everything?
Some participants cheat, right?
Yeah.
So they actually do a lot of data mining into papers so that they can automatically annotate information that's already there. But hey, if you can do that well, I'm very happy, right? Because that's a pretty hard problem.